Hello, everybody. Hello, everybody. Oh, I'm too tall, I guess. My name is Stefan Braam, founder and CTO of Cellistic. I'm going to talk to you about our off-the-shelf cell therapy platforms to accelerate allogeneic cell therapy. So the founding thought of the creation of Cellistic was the recognition that you therapeutic developers have created fantastic technology that now actually cures patients. So we have multiple approved drugs, but some industry challenges remain. So we see with autologous cell therapy that even the largest facilities in this world can maybe address 5,000 patients a year. Thanks to all the great work, we see a growing number of indications. So I think there is a lot of good points that we are moving outside hematology into solid tumors and maybe even in other indications. And clinical data justifies a much earlier line of treatment, so the number of eligible patients will go up. So the manufacturing costs are high, and I don't think there is a real foresight or indication that it will go down. And as a result, we have reimbursement challenges in the US, but definitely in other parts of the world. So industry has responded typically with the question, how do we scale this? So a number of cell therapy companies have made the step from autologous to donor-derived allogeneic. And that is still quite a similar manufacturing process. We now start with donors, and we start to generate larger batches. That addresses scalability, that addresses manufacturability to a certain extent. Quality and characterization can go up because you start to cryopreserve. The price might go down and repeat dosing becomes possible. But as you can see, the Harvey balls are not completely filled here. So there is significant room for further improvement to actually go to that product-specific therapy that we want to make. So the reason that we strongly believe that iPSC-derived allogeneic is a better approach is starting with the bottom. What you see with donor-derived allogeneic is that you run into exhaustion, and exhaustion limits your scale-up and dose capacity. With an iPSC-derived cell, you can actually scale at the iPSC stage, at an HSC stage, at a progenitor stage, at a final stage. So your ability to scale is much and much larger. Your manufacturing workflow is actually looking very different because you are going to introduce your gene edits not at the end of the process leading to high vector cost, but you do it at a master cell bank. And in fact, you do it once and you fully characterize that cell, you make that clonal. So we see now concerns popping up. There was a beautiful paper in Cell talking about CRISPR in T cells, that T cells actually losing chromosomes, quite scary. Here, you actually test that cell, you make sure that the cell is fine, and then you go forward. The final point is that in donor-derived allogeneic, you typically see that you need multiple donors because you cannot scale so far. So you get a lot of comparability challenges in your preclinical development, but also in your clinical development. You're going to generate a super batch and you're going to blame the donor. And that is obviously not very helpful, leading to very complex clinical trial designs. So how we want to respond to this as Cellistic is to actually build iPSC manufacturing platforms and make that available to therapeutic developers. So there are two manufacturing platforms. One is called Pulse. It's for cell line development. What we essentially do is to go from ID to edited and banked iPSC, incorporating all the features that make a healthy cell a product. And the second platform is called Echo, where we differentiate the iPSC to the drug product. So the cell types that we're currently working on are NK cells, alpha beta T cells, gamma delta T cells, and macrophages, so very focused on the immune space. And with that, we simplify, we de-risk, we speed up the development of these novel therapeutics. So from a manufacturing workflow perspective, it looks like this. So Pulse covers donor material, reprogramming, and editing. Starting with a donor on the left, we reprogram, we make an iPSC, we bring in with our CRISPR technologies product-specific features, we create working cell banks and master cell banks, and then they are subsequently being manufactured towards the drug product. So in Pulse, what we are doing, I mentioned reprogramming, gene editing, and cell banking. So this becomes the basis of the product. So what is very important is to be quick, to be efficient, and to do this at a high quality standard. What we are seeing right now is that certain therapeutic developers have actually pushed cells into the clinic that have been generated in R&D labs. We are doing it different. We have built a lot of automation, a lot of workflows. It's fully overseen by a quality assurance team, high quality materials, batch records that are being reviewed by quality assurance, and a lot of analytical testing to provide confidence in the cell that is being generated. So in terms of performance, we've selected our nuclease of choice. With that nuclease, we can edit at high efficiency, single edits, we can duplex edits, we can do triplex edits. And remember, we are doing a clonal selection, so even if we have 30 to 60% of editing efficiency, testing 10 clones will be more than enough to find the right cell. Then to provide the assurance that we're actually making the right cell, we spend a lot of time on clonality assurance and healthy cells. So we can clone now with very high cloning efficiency. We make sure that these cells are absolutely clonal by imaging at a nozzle and imaging from the bottom, following these cells grow over time to provide confidence in that part of the process. So from there on, we move into the manufacturing process. I mentioned NK cells, alpha beta T cells, gamma delta T cells. And so I think what is unique is that we have brought together very flexible, state-of-the-art process development labs where we have all the manufacturing technology. Next door, we have a GMP facility that we actually acquired from Celiad Oncology last year. We also brought in their complete teams for QC production, and quality assurance. And so with that team, we brought into the company the experience of 200 plus clinical phase 1, 2, and 3 batches into the company. So on top of that, we've now built all the cell type experience for in-process control and QC testing. So in terms of the facility, it's completely redeveloped and remodeled. It's mechanical completion in December this year. So it's designed in such a way that we can do open operation steps, that we can do bioreactor steps in grade C. We are only using off-the-shelf manufacturing technology, so no weirdness there. And we ensure that we have an optimal flow where we can manufacture, where we can do the cryopreservation, visual inspection, etc. So in terms of a manufacturing process, so this is an NK example. What you see are well-defined unit operations, starting with iPSC on the left, that we differentiate in HSC, that we commit to the lymphoid lineage, that we commit to NK. If you look at the expansion, so we actually in that process expand 30,000-fold. So you actually only need a few iPSC cells to go to our target batch size of 75 billion cells. So that's actually less than a T25 of iPSC. So the expansion really happens during the process. This process is built completely feeder-free. No unscalable cell sorting steps, chemically defined media. So from day one, my teams are really focusing on that manufacturability to get to the cells that we want to make. We ensure that that process works across different lines so that it's not line specific. And in terms of characterization, so an NK, natural killer cell, is actually active without any product-specific features. So if we mix that with tumor cells, they actually kill. So within 4 hours, the majority of the tumor cells are dead. When we look at the cytokines secreted by those cells, we see interferon gamma, we see TNF-alpha as expected, we see GM-CSF, which should recruit macrophages, and we see the killing enzymes performing in granzyme B. So we actually make the right cells that actually look like the NK cells that you want to make. So in summary, what we are doing is enabling therapeutic developers a new path towards iPSC allogeneic cell therapy. So there are obviously a couple of great companies out there that have built everything themselves, the targeting of the cell line development, the manufacturing process. And so what you see if you analyze that, and this is all public data, that they have spent more than 5 years and more than 100 million to go to an IND. So that's a lot of money, and especially in this economic climate, it's probably not so feasible. What we are doing through our platforms is to greatly accelerate that process and also greatly reducing the cost because you can work from pre-established platforms. So in summary, I went through the advantages of iPSC as a starting material, which are, in our opinion, the optimal way of doing this. There is this clear requirement for allogeneic cell therapy manufacturing where the process is still very important and where the process still impacts the product. So you want to lock that down as early as possible to be as efficient as possible. And so therefore we have built Cellistic platforms that expedite that product development, avoid common part files, that exploit pre-established scalable processes with predictable cost, and brings that standardization that you want to have in preclinical development from day zero. And with that, I end. So I'm the tall guy, so if you want to talk to me, you can always find by looking for the tallest guy in the room. Happy to talk.